Good afternoon, ladies and gentlemen. Very happy to stand here again. And this time I will show some posters and oral presentation about embryology and embryo metabolism. And in our clinical work, we often use a growth factor in some patients with poor oocyte quality or older age. And we're just thinking about if we can add some growth factor in the medium to help them. So this study from United Kingdom about the effect of GM-CSF on muline embryo development, aneuploidy DNA fragmentation, and morphokinetic parameters analyzed using embryoscope. So this study, some studies have shown that GM-CSF enhances blastocyst development. Others have shown no effect. And two studies have shown that GM-CSF have no effect on aneuploidy in human and muline embryos. And fortunately, in 2011, a culture media was launched containing 2 nanograms per milliliter of GM-CSF, and it claiming that it can enhance blastocyst development. So this study, using the culture medium from the 2 cell stage muline embryos cultured singly in the embryoscope until blastocyst stage, and in the presence of different concentrations of GM-CSF. Let us look at the timings and according to the GM-CSF different concentrations from 0, the control group, and 1, 2, 5, 10. And the muline blastocyst development decreased with GM-CSF versus control group, which is significantly even in the last group, 10 nanogram per milliliter group. And about the aneuploidy and mosaicism, according to the different concentrations of GM-CSF, we can see that there's no significant difference in aneuploidy mosaicism between control and the GM-CSF group. And about the cell number with DNA fragmentation, we can see that there's no significant difference in cell numbers with DNA fragmentation for control, but only in the proportion of nuclear with DNA fragmentation in 1 nanogram per milliliter group and 5 nanogram per milliliter group. So it is difficult to explain. So conclusions were at high concentrations of GM-CSF, a significant negative impact was observed on the blastulation rate, and no effect of GM-CSF was observed on aneuploidy or DNA fragmentation. More supporting evidence is required to ensure that the efficiency and the risks of growth factor adding in the media. And the mouse embryo is not an ideal system to study human pre-implantation development. So next study is about a human embryo. This from Greece, about the effect of granulocyte macrophage colony stimulating factor GM-CSF on pregnancy rates in patients with multiple unsuccessful IVF attempts. So you have already know about GM-CSF that can promote blastocyst formation and to reduce inner cell mass apoptosis, as well as to play a vital role in placenta development and fetal growth. However, it is still unclear whether multiple unsuccessful IVF attempts should be another indication for the use of GM-CSF. So this is a prospective observational study in patients with more than 2 times previous unsuccessful IVF attempts. Oocytes were fertilized, cultured, and transferred in embryogene medium and containing 2 nanogram per milliliter. And in another group is the lutein and the control lutein medium. So in both groups, embryo transfer performed on day 2 or day 3. And about the stimulating protocol and the number of follicles on day of HCG and estradiol level on day of HCG injection and progesterone on day of HCG ingestion. They are comparable between the two groups. And about embryological data is also comparable between the groups. Let's focus on the clinical pregnancy and implantation rates. You can see that a clinical pregnancy rate, the second column, you can see that in the group 1, with the higher clinical pregnancy rate, reached to 38.2%, and the control group is 22%, is higher in clinically, but did not reach the statistic significant. And implantation rate is same. In the study group was higher about the implantation rate, 19%, but in the control group, 11%. And also a study to evaluate the effect of GM-CSF according to the age. They divided the two groups according to the age, less than 39 and more than 39 years old. And the great effect can see in patients aged younger, but no, the difference is not statistically significant. So the study concluded that ovarian stimulation data and embryological data are comparable between groups, and the clinical pregnancy rates and implantation rates were higher, but did not reach statistical significance. And inclusion of GM-CSF in embryo culture media may improve the pregnancy and implantation rates in patients with multiple unsuccessful IVF attempts. So the RCT studies still lead to evaluate the benefit of using and also the risks. And this study is from Japan about the outcome of breast cyst vitrification with cryoloops. So the question is ultra-rapid vitrification method of breast cyst using a cryoloop safe and effective because it's open the vitrification, the freezing? This study is a retrospective analysis including 2,924 vitrified thawed breast cyst cycles, and the control group is 2,003 fresh breast cyst cycle in women aged less than 40 years old. And IVF and ESHRE procedure carried out in sequential media, and breast cyst vitrified using cryoloop technique and vitriblast. And about implantation and congenital abnormality rates. And besides this, we firstly to look at the mean number of embryo transfer. The difference have the significant, statistic significant. In the fresh cycles, a little bit higher, the more numbers of embryo for transfer. And the clinical pregnancy rate and the delivery rate and average birth weight are all higher in the fresh cycle. But the key point of IVF cycle is implantation rate to evaluate the embryo per embryo quality. So the implantation rate in study group is 39, and the fresh cycle is 38%. So there's no difference. So conclude that the ultra-rapid cryoloop vitrification method of breast cyst is easy to use, safe, and effective. Outcome of ultra-rapid cryoloop vitrification method of breast cyst is comparable with that of fresh breast transfer, and in terms of the implantation rate and the congenital abnormality rate. But we still need long-term follow-up of children from ultra-rapid vitrification method of breast cyst using a cryoloop, including motor and psychological development. I think it's also include other new technologies in IVF technologies. This study also from Japan about the restoration of developmental competence of oxidatively damaged oocytes by meiotic spindle transfer. So, as we know, oxidative stress during oocyte maturation might contribute to age-related oocyte aneuploidy and infertility in the human. So, the ability of ooplasmic replenishment by spindle transfer at a metaphase 1 stage to treat oxidatively damaged oocytes had not yet been assessed. So this is a method. You can see that the first, the one group, the oocytes be treated with H2O2 to cause the oxidative stress. And another group is intact oocytes. This is a control group. And from the two groups, they isolated the spindle from the H2O2 treated oocytes and transfer to the intact oocytes. And also isolated the spindle from the intact oocytes and transferred into the treated oocytes. So we have the 4 groups. So group A, group B, and group C, and group D. So let's compare with these 4 groups. In the group, H2O2 led to significantly lower rate of mature from M1 to M2, and shortened spindle length in oocytes reached M2, and the lower MMP in matured oocytes. And in the group, the no H2O2 treated oocytes, this is a control group, developed blastocyst stage to 40%. And following spindle transfer in the C and D, and following spindle transfer, we can find that restore the rate of maturation to M2. So normal M2 spindle morphology and increased MMP in matured oocytes and higher rate of the development to blastocyst stage. So quite interesting and promising result. So So this concluded that oxidative stress impaired meiotic and mitotic development of mammalian oocytes with reduced mitochondrial activity and aberrant spindle formation, and sometimes in the older patients with age. So such plasmic damage can be reversed by replenishment of ooplasm, indicating that spindle transfer can play an important role in understanding age-related oocyte pathology. So this study also have limitation. This study was conducted using a mouse model with artificially induced oxidative stress, so the findings may not directly represent human infertility. And this study from Spain about impact of exposure to music during in vitro control on embryo development is quite interesting and full of imagination. So as we know, it has been reported that exposure of in vitro culture human embryos to 5-second intervals, 44 hertz of hour of micro vibrations can improve embryo development and quality. This is the laboratory data. But nothing is known about music as a source of mechanical vibrations and its effect on human embryos whilst undergoing in vitro fertilization. So in this study, 985 oocytes from 114 patients were analyzed. Inseminated oocytes from each patient were randomized, divided into groups, cultured with music and cultured without music. And also they selected the 3 types of music: pop, heavy, mental, and classical, because we don't know which kind of music the embryo like. The fertilization rate and the embryo quality were compared between the 2 groups. Now, about the fertilization rates, the blue column is with music, is 76%, and without music, only 71%. But we compare use the statistics software, the difference is statistically significant. About embryo quality, no statistically significant difference was observed between two groups. So the routine use of music inside the incubation or inside our clinic, it probably will improve fertilization rates, does not affect of embryo quality and calculated according to the presence of multinucleation cleavage stage of embryos. So currently, more studies needed. So this study from Greece about metabolic profile of human embryo and its association with chromosomal abnormalities. And this study is to check the metabolic profiling. The embryos subjected to day 3 biopsy were proceeded for pre-implantation genetic diagnosis. And they were, after biopsy, and before biopsy and after biopsy, collected the culture medium for test the metabolic profile using gas chromatography mass spectrometry, GCMS. And in day 3 spent culture media, the levels of lactic acid, aspartic acid, and glycine significantly higher in chromosomally normal and versus chromosomally abnormal embryos, and significantly lower concentrations of pyruvic acid, glycine, and glutamine for chaotic embryos compared with uniformly aneuploid embryos. So the key point is chromosomally abnormal embryos show characteristic patterns of bimoleculars in spent culture media on day 3. So how about on the day 5 spent culture media? See, we can see analysis of day 5 spent culture media also revealed differences in levels of aspartic acid, glutamic acid, and methionine, and lysine and tyrosine, and cysteine between chromosomally normal and abnormal embryos. But we don't know if which one is the cause and which one is the result. Probably the abnormal metabolic profiling causing abnormal chromosome? I don't know. So what's your opinion? What's your comment? So also in this study, they confirm the all embryos, again, the rejected embryo with the using the PGS and PGD about the aneuploidy. So this study concluded that provide characteristic patterns of biomolecules found in day 3 and day 5 spent media from embryo, which were identified as chromosomally abnormal following PGD and PGS. And difference were observed between uniformly aneuploid and chaotic embryos. And this study, the limitation is normal embryos transferred on the basis of single cell analysis, but not resulting in a pregnancy could not obviously be tested for mosaicism. And this study is from Denmark about cumulus cells and gene expression markers for oocyte competence. Yes, in our work, it's difficult to predict the oocyte competence. So the aim of this study was to describe work on quality classification of gene expression profiles in correspondent cumulus and granulous cells. As you know, a number of studies have already identified genes associated with developmental competence of oocyte. And however, there are no consensus in suggested marker gene, no common standard for oocyte competence, and differences isolation technique. So there's so many genes, we don't know which kind, or is like to look for the needle in the ocean. So in this study, they included 16 children born following IVF-XC treatment, and they included in the 16 follicles, the cumulus cells, and 16 age and 16 morphology matched Hcg negative follicles. These are pregnant and another group is non-pregnant. So they take out the cumulus cells from the follicles and extraction of RNA and to analyze the gene expression. And the 48 gene signature identified that best separated the pregnant and non-pregnant expression profile. And the classifier was 84% successful in predicting pregnancy using 32 expression profile, too much. The classifier was successfully validated in an external data set, and the success rate is 92%. So the ideal marker of oocyte competence should include the following characteristics. It is objective, it is clear limit between good or not, and easy and fast to use, and cheap and non-invasive. But unfortunately, we are now get closer, but we're not yet at this stage. So we still need more studies and more long way to go. This study from France about reverse phase protein array, this is a new approach for the identification of cumulus cells by markers of oocyte quality. So it is a good one. So the aim of this study was to assess the use of reverse phase protein array. This is a method to detect and analyze the potential biomarkers related to oocyte developmental competence through a non-invasive procedure involving each individual cumulus cells in human. RPPA is a sensitive and quantitative technique that allows the detection of specific proteins in very low quantities of biological samples to help clinical management of cancer. So can we use it in infertility area? So in this study, the antibody firstly isolate the cells and created at a high density of antibodies, VCL, ERK2, and SRC on the slides, and then using the fluorescent to check to detect the gene, the signal of the gene, the expression. So, and then they use a Western blot to validate the protein. So 13 individual cumulus cells collected from 4 ICSI patients and a pool of 16 cumulus cells from 2 patients used to define sensitivity and specificity of RPPA. So in 4 patients, all the cumulus cells, VCL, ERK2, and SRC, were detected in all analyzed individual cumulus cells. So the key point is RPPA with near-infrared dyes can specifically detect and quantify proteins in individual human cumulus cells. So probably we could use this method to help us in our research. And this study's limitation is antibody validation remains a challenging problem. This study also from France about maternal age affects angiogenic factors and insulin signaling pathway of human cumulus cells isolated from mature M2 oocytes. The impact of maternal aging on the energy metabolism and the post-transcriptional process of human cumulus cells are previously reported. However, little is known about the angiogenic factors and insulin signaling pathway if the maternal aging have the bad or worse effect. So the gene expression profile of cumulus cells from mature oocytes collected from patients aged less than 30 years old were compared with those from patients older than 30 years old. So, but this is not older. Probably should divide and divided the patients with 35 years old or 38 years old. The human genome plus microarray was used. All cumular cells from each mature oocyte were collected. These are two groups from the patients aged less than 30 and more than 30, and RNA extraction and amplification, and then using microarray. You can see that the red color corresponds to the upregulated genes, and you can see the difference in the group 1, the top genes groups, the upregulated, but in another group, the downregulated. So molecular signature of human cumular cells is affected by maternal age according to this result. But further investigation still needed with a larger number of patients to confirm these results. And this study from China, from Lijie, the first affiliated hospital of Sun Yat-sen University, and about the embryonic human chorionic gonadotrophin, HCG, in spent culture media may tell embryo viability in IVF-ET. And this is a multi-center study. And HCG is very easy to test and monitor in our clinical work. So how can it tell us about embryo quality and help us to improve the outcome of IVF? This study is a cross-sectional study in 6 IVF centers from November 2011 to October 2012 in China. And a sequential culture system was employed from 2 p.m. to blastocyst stage. We can see that the higher beta HCG concentration showed in fresh blastocyst with expanded cavity or higher trophectoderm grading. So the difference is significant. A significantly higher beta HCG level emerged in pregnant women, both on day 3 or day 5. And in the concentration of beta HCG correlated positively with implantation rate in fresh day 3 and day 5. These are all positive results. And the last one is not. The higher beta HCG concentration was found in ongoing pregnancy rate than in miscarriage in fresh day 5, but did not reach, it also reached the statistical significance. And the concentration of beta HCG correlated positively with implantation rate at day 3. So the study concluded that higher embryonic beta HCG level was found in embryos higher morphological grading or implantation potential. And HCG testing is so easy, so probably we should repeat this study and to evaluate if it is valuable to help us to select the good quality embryos. This study limitation is a cross-section study and not a randomized control study. So thank you very much for your attention.
Thank you very much, Professor Ma. It's not very easy because we have 14 abstracts in this session. Yeah, great effort. Yeah. I like that music stuff very much.
Yes.
If I'm embryo, I prefer jazz.
Yes.
Right? Okay.
Yes.
Okay, is there any question or comment from the floor?
Yes, we also can use the time-lapse imaging system to monitor if when they listen to the music, can they dance or develop quickly.
Yeah. Well, that's ESHRE.
Yeah, most life
Okay.
love beautiful things.
Yeah. Any further comments? How about a friend from Japan? I remember their clinic of Morimoto, they have a lot of music in the Morimoto clinic, right? Yes.
Dr. Chang from
Yes.
Taiwan. And from your last session, last topic you reviewed, and we found embryo had Hcg secretion. So where do you think it's from?
Secreted.
You found the culture
HCG.
medium inside, you found Hcg content. So where do you think it's from?
And from embryo, the embryo.
So embryo will have HCG at that time. So you think embryo will secrete HCG at that time?
So I
And that
cannot answer
stage?
that.
Yeah, probably.
Yes.
Yeah, probably because.
But we
Yeah.
detected the existence of the HCG. In the media, only embryo live there. So who can secrete?
I think remember about, I think every bonsai in Singapore, they have every done a study. The HCG is a marker of a processes. So it's probably a transcript the HCG on the day 6, just reaching the hatching stage. They will secrete some HCG. So I remember Dr. Mao have a report, a paper
Yes.
in this morning session
Yeah.
talking about the divided concentration of HCG, and it's a positive correlated with the pregnancy outcome according to HCG, right? The medium level.
Yeah.
Okay.
They're quite interesting question.
Yeah.
So we should, HCG is easy to detect.
Yes.
So we should perform more studies about this.
Further comment? Okay, we need another break again, right? Okay. And we thank them all
Thank
again.
you very much.
Abuse.